Hello students, welcome to a video class on Metallurgy Lab by the Department of Mechanical Engineering, TKMCE. Today we will study how to identify the material of the given specimens by metallography. Metallography is the study of the structure of metals and metal alloys through the examination of specimens with a metallurgical microscope. The structures observed in the microscope are often recorded photographically. First, we have to make a brief study on the metallurgical microscope. A metallurgical microscope is like any other optical microscope with the exemption of the lighting orientation. A metallurgical microscope uses a different lighting method that can illuminate solid specimens to identify, inspect, and measure them. A conventional microscope illuminates a transparent specimen from below the stage, making it visible through the eyepiece. Since light cannot penetrate opaque or solid objects, this is not a suitable method for observing these samples under magnification. Metallurgical microscopes illuminates objects using polarized light with an external light source. The microscopes are categorized as upright and inverted on the basis of the position of objectives relative to the stage on which the sample is placed. In the upright microscopes, the objectives are placed on the top of the stage. In this type of microscopes, the size of the sample is limited to an average height of 80 mm. On the other hand, in the inverted microscopes, the objectives are placed below the stage. So if you work with big and heavy samples, inverted microscopes provide you the freedom you need. Inverted metallurgical microscope has the objective under the stage, which allows viewing the specimens at varying sizes. We use an inverted metallurgical microscope in our lab. Microscope parts and functions. The eyepiece is the lens of the microscope closest to the eye that you look through. There is an eyepiece holder which simply connects the eyepiece to the microscope body. We can easily change the eyepiece to vary the magnifying power. The body is the main structural support of the microscope, which connects the lens apparatus to the base. The nosepiece connects the objective lens to the microscope body. With a turret or rotating nosepiece, as many as 5 objectives can be attached to create different powers of magnification when rotated into position and used with the existing eyepiece. The objective is the lens closest to the object being viewed, which creates a magnified image in the area called the primary image plane. The focusing mechanism has the adjustment knobs to allow coarse or fine variations in the focusing of the stage or objective lens of the microscope. The platform on which the prepared slide or object to be viewed is placed, we call as the stage. The means employed to light the object to be viewed is the illumination source, and many microscopes have the electrical light source for easier and more consistent lighting. The bottom or the stand upon which the entire microscope rests or is connected is called the base. There is a digital camera which captures still images and video and allows you to view a live stream on the attached computer. Now we can go through how to prepare a specimen. The technique of preparing test specimen can be divided into 2 groups, grinding and polishing. First, we have to select the specimen, select a suitable surface, and using a simple hand file, you can prepare a level surface. The hand lapping includes 5 or 6 graded abrasive papers to produce a sequentially finer surface finish. Start with a coarse grit of 100 and using a firm and uniform pressure, slowly move the specimen forward and back across the abrasive. This will produce parallel scratches of uniform size. Continue this step until the entire surface of the specimen is flat and contains only scratches of the size of 100 grit abrasive. When the sample is flat and the only scratches remaining are those due to the 100 grit size, move to the next paper, say 200 grit size. The direction of grinding is changed from paper to paper so that the removal of the previous grinding marks is easily observed. Repeat this procedure for different papers of grid size varying from 100, 200, 300, 400, 500 and 600. Checking after each step to be sure that only those scratches remain that are due to the smallest grit. After completing the grinding operation, the same process is repeated through a set of polishing papers ranging from 0 to 4. Now we have a surface with much better surface finish. Let us now move to the polishing wheel. Polishing is carried out on a cloth-covered rotating wheel covered with a suitable pad that is impregnated with either a suspension of polishing alumina in water or a diamond dust oil. Let us now see how the alumina powder or the diamond cut compound is used for polishing. Water can allow to slowly drip during the polishing operation. During the polishing, the specimen should be held firmly in contact with the polishing wheel, rotated or moved around the wheel to keep an even polish. The specimens must be washed and dried after the polishing steps. So these are the two specimens which we have prepared. You can see the polished, clean and dry surface which is now ready for the etching process. In most cases, an etching reagent is needed to develop the structure of a metal so that the structure can be examined with a metallurgical microscope. Here, we use nitrile as the etchant. Nitrile is a solution of 2% nitric acid and 98% methanol. A small amount of the etchant solution is prepared and poured into a small petri dish. The polished sample is then immersed in the solution for approximately 5 seconds. After etching, the sample is washed and dried. Drying is usually carried out with a hand dryer. Now, our specimen is ready for metallographic observation. Now let us see how we can observe the metallographic microstructure. Place the specimen on the stage of the metallurgical microscope connected with the computer. Adjust the magnification, focus and positions, and then we will get its microstructure on the screen as well as on the eyepiece. Here we can see the images obtained from the first specimen at various magnifications. By analyzing these images, we can understand that the given specimen is cast iron. As you know, the cast iron microstructure is characterized by the presence of graphite flakes in a ferrite or perlitic matrix. Similarly, these images can be obtained for the second specimen also. From the microstructure, we can confirm that the second specimen is mild steel. And as you all know, the mild steel microstructure is characterized by the presence of ferrite and perlitic microstructure, and we can see that the perlite islands evenly distributed in a ferrite matrix. Thus, we have understood how to identify the metallographic microstructure of the given specimen. Thank you.